The most common method of DNA sequencing involves a modified DNA synthesis reaction. The reaction mixture contains many copies of the segment of DNA to be sequenced, as well as reaction primers and plenty of the four types of deoxynucleotides, or DNTPs, to be used as building blocks in DNA synthesis. This sequencing method also requires so-called chain-terminating dideoxynucleotides, or ddNTPs, in the DNA polymerase reaction. Whereas DNTPs have a hydroxyl group at the 3' position of the sugar, ddNTPs lack the hydroxyl group. The hydroxyl group is required for chain elongation, so ddNTPs are considered chain-terminating nucleotides. 4 different types of ddNTPs are added in small amounts to the reaction, with each carrying a different colored fluorescent tag. As DNA polymerase adds nucleotides to the growing strands, a random choice determines each step. Will a DNTP bind, letting the strand continue to grow, or will a ddNTP bind and terminate chain elongation? As the process continues, many incomplete DNA strands form, with a ddNTP as the last nucleotide incorporated. The size of each fragment is determined by its terminal dideoxynucleotide, marked by a specific color fluorescence. A relatively long strand that fluoresces blue identifies the position of a cytosine-bearing nucleotide on the template strand near the 5' end. Each length and color identify the position and type of nucleotide in the template strand. The 4 newly synthesized strands shown here would be only a small portion of the strands synthesized in the entire reaction mixture. The last incorporated nucleotides in these fragments provide the complementary sequence to the template DNA strand. This complementary sequence is GAA, CAG, TTG, CAT, CAG. And therefore, the template strand consists of the sequence CTG, ATG, CAA, CTG, TTC. How do investigators put the fragments in the correct order? Through gel electrophoresis, the mixture of fragments can be ordered according to size, with the smallest fragments migrating the fastest in the gel. An automated device scans the gel. A laser beam strikes the bands on the gel, causing the tags on the DNA to fluoresce their unique colors. A computer saves the series of fluorescent colors and determines the corresponding sequence of the DNA.